complex genomes, for example, the human genome, and provides a shorter sample preparation time relative to the TrueSeq PCR-free and Nano library prep kits. The protocol is similar to the TrueSeq Nano DNA library prep kit, although with a few differences. Unlike the TrueSeq kits, fragmentation and adapter ligation of genomic DNA, also known as tag mutation, occur in the first step. This is done with an enzyme called a transposon, which is a transposase-transposon complex. The enzyme is able to make cuts in DNA like a transposase, but also fidelity NGS data. It utilizes a high-fidelity DNA-dependent polymerase to generate a complementary copy to a single-stranded DNA template. In each reaction, a single primer, complementary to the template, initiates a DNA synthesis reaction from the 3' end. Deoxynucleotides or simply nucleotides are added one after the other in a template-dependent manner. Each reaction also contains a mixture of 4 dideoxynucleotides, one for each DNA base. These dideoxynucleotides resemble the DNA monomers enough to allow incorporation into the growing strand. However, they differ from natural deoxynucleotides in 2 ways. 1, they lack the 3' hydroxyl group, which is required for further DNA extension, resulting in chain termination once incorporated in the DNA molecule. And 2,
washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the
hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters.
Humanity has a long history of figuring out ways to modify genes. We have been crossbreeding plants to make them better to eat for thousands of years. But with gene editing, we've made a great leap forward. This increasingly accurate new technology is faster to use, relatively simple, and cheap compared with previous methods, and promises huge benefits. In a nutshell, it works by identifying, then cutting pieces of DNA. One way of doing this uses a component known as CRISPR to pinpoint the precise DNA sequence within the gene to be altered, or allowing it to be replaced by another stretch of DNA that is introduced at the same time. This can either replace a faulty gene with a healthy one, or change a gene to make it behave differently. The methods act like a find and replace for the genetic instruction manual. By making these microscopic changes to DNA, gene editing has the potential to make big changes to our lives.
Together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its 2 nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, The fixing process is error-prone and often inadvertently introduces
control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA.
Surgery. When Lulu and Lana was just a single cell, this surgery removed the doorway through which HIV entered to infect people. A few days later, before returning Lulu and Lana to Greece ward, we checked how the gene surgery went by whole genome sequencing. The result indicated that the surgery worked safely as intended. Grace's pregnancy was normal, which we monitored closely by ultrasound and blood tests. After birth, we again deep sequencing Lulu and Lana's whole genome.